I might kind of kick off on a few questions. So one that's come in from the online was a question prompted by Siddharthan's comment about the use of wearables and smart devices, and asking whether they are being used in UK Biobank, and apart from the accelerometer, they aren't. But I wondered whether you or other members of the panel might say, what device might we use either in this dementia assessment that Paul just mentioned, or maybe even more broadly across the UK Biobank population?
I could start. I mean, the reality is lots of people are now using various apps on their phones, on their finger, to measure elements of their behavior, whether it's sleep, whether it's steps, physiological. Lots of people are working with the Zoe app. So you might think of cognitive games. This is a very crowded space. Your voice is another potential high-frequency data set that you could acquire, which likely will segregate different conditions. So you can imagine on a phone-based app, a combination of some cognitive games, including visual inspection, time, your voice, physiological measures, movement, gait. That alone, if you can do it at scale, and you've got a measurement of the truth, which is why you need these longitudinal, deep, clinically phenotyped cohorts, will be very powerful. But if you then layer into that blood-based, which is just dry blood spot, and or genetics, you're at the races. You can transform things. And just to be clear, all of this is going to happen in the next 10 years. The only question is whether the UK is going to play a role in this. And heaven knows the UK has enough problems, but one of our assets are cohorts, world-class science, biobank, and all these other initiatives. What we're trying to do is bring this all together.
Well, I'd add to that, we have extraordinary altruism in this country. In the 500,000 participants in UK Biobank, coming to the imaging visit, which is a 5.5-hour visit, and then having gone through that, over 70% are willing to come back and do it again. So do you think exploring with participants their willingness to have an app on their phone that would monitor things like speech and how they use the phone, how they leave their homes and things like that?
So I think no question we should do that. We also need to think through, because of the particularities of the UK Biobank consent, how we feed back information to participants or their primary care physicians, because some of this information is now actionable. And so that needs to, and I know you and Naomi and others are thinking about it, but this is an open goal. And we've got many shots on goal. We mustn't be like the Scottish football team. We need to put the ball in the back of the net.
So maybe I can ask Vanessa, you've heard the different talks, and I was just kind of wondering whether you wanted to expand on that or any other kind of aspects of what you've heard in terms of, I mean, one of the things I find most useful is when people come and talk to us and say, UK Biobank is useless for the kind of research I want to do, you should do this. And I think that's what prompted us to think about dementia. You know, we were quite clear that the dementia information we had was inadequate. But your comments?
Yeah, absolutely. And I think, as has been pointed out by the speakers, we're at an immense time of opportunity, both in terms of disease-modifying drugs really being available for Alzheimer's dementia for the first time, and scalable biomarkers that would allow us to identify people. But the reality, as has also been pointed out, is that we struggle to identify people. We struggle to diagnose them accurately. We struggle to identify even people with earlier levels of disease. And part of that's because as the sheer numbers have increased, most of these people don't even necessarily sit within memory clinics where they can be assessed even to the level that we've described is inadequate, but they won't even get that. Most people sit in the community or in primary care. So the other opportunity for things like the digital biomarkers is not just identifying people and accurately diagnosing them. And I would say, you know, as somebody who's been working in memory clinics for kind of almost 30 years now, there's been a real shift in the last few years. We used to think that people didn't want to know about an accurate diagnosis while there wasn't a disease-modifying treatment. That is changing. I think people recognize the validity of understanding their diagnosis and how that enables them to plan, manage lifestyle choices, etc., whether or not we have a licensed treatment. But of course, if we're going to institute these treatments, the other thing that we lack data on is the trajectory of disease once we diagnose it. And because we don't see people in secondary care, we're limited in what data we can collect. So another real opportunity around the kind of digital and scalable biomarkers is to understand the trajectory of people's illness, how they respond to disease-modifying drugs, how they respond to personalized treatment plans, risk management, etc. And how best you target the services that you do have available to those people. So I think there's a huge wish to engage from the public and recognizing that we need to shift services, and these biomarkers present an opportunity. But we need good ground data, and that's where things like UK Biobank, I think, come in.
Before I go to Paul, can I just ask you, Vanessa, from your experience, just following up on that, do you think if we asked UK Biobank participants who have dementia to come to such a clinic that there's likely to be kind of a positive response to that?
Yeah, absolutely. I mean, again, over the years, I've always been blown away, to be honest, by people's willingness to undergo huge breadths of assessment, sometimes quite unpleasant assessments, to be honest, but because they understand the urgent need for this data. And as I said, that's just increasing all the time. So I think, again, you'd be pushing an open door in terms of participants, and then in terms of thinking about maybe long-term follow-up as well with similar biomarkers. I think people would be very willing.
Okay, Paul, make a comment, and then there are a couple of questions just down here.
Yeah, no, thanks. I just wanted to follow up on the comments to emphasize why we're talking about this in a UK Biobank meeting. So the points that Siddharthan made are absolutely right. The things that he described are going to happen, and they are happening in the external world. What is unique about the discussion we're having today is to make the point, derives from the fact that in order for these tools in the outside world to be useful, to be accepted as veridical measures of disease progression, and to be accepted by regulators for the approval of new medicines, is that they be established as valid. And the UK Biobank population, where we can make controlled observations that then can be correlated with the remote observations, provides a unique platform for this. It is also unique because I want to highlight too, that while the brain health clinics that Vanessa is describing are starting to make a real inroads into the space, they're still taking in a different population, a later population, than the one that we want to be treating, which is that early and, in fact, the preclinical disease. That's where UK Biobank, again, can make a truly unique contribution to the ecosystem.
Thank you. So there's a question just down here and then one here.
Dave Curtis, UCL. More of a comment, really. I'm really disappointed at this. I mean, that we can invest in getting every single participant whole genome sequenced, and we can't find out which participants have got dementia and haven't got dementia. You know, step number one. And then to go on to get the A beta, the p-tau, that, you know, to establish people who've got Alzheimer's disease, the way people talk about, the last speaker didn't do this, but so many people talking about dementia as if it's a diagnosis, and it's like saying shortness of breath is a diagnosis. We've got to know who's got Alzheimer's disease, we've got to know who's got cerebrovascular disease, which is quite treatable and preventable, and we want to know, we need to properly invest in this unique resource and actually find everybody who's got Alzheimer's disease, and actually, study those, not a small sample of them, but all of them.
Siddharthan.
I think it was Dave. Dave, we all agree with you. You should come and write the grant to get the money. So there's no question. Everything you've said is correct. But that's why we're here, because we're going to fix that.
Yeah, I mean, as I said, what we're looking for are not, is what's wrong with UK Biobank and how we can make it better. And that's exactly what we want to do here. There's a question just there to your...
Yeah. Elijah Bear from St George's, London. I've noted that the death data gives a significant proportion of your outcomes for dementia. It's also, I note that the pathology of the death data may be the things that give you the granularity that others are searching for. And therefore, the outcomes in terms of death data are going to be very important for prediction from Biobank, if that's feasible, at least for a subset that may be extrapolated to others. Is there going to be an opportunity to revisit the linkage of pathology data, autopsy data, coronial data that we've discussed in the past in Biobank? I know it's a real stretch from the legal perspective, and Naomi and Kathy and I have gone through this many times, but it could also be relevant to other phenotypes other than dementia, of course.
Yeah, it's a really good question. I mean, to be honest, the problem with access into coroner's reports is there's no central place where these data are stored. So you have to go to each and every coroner's office up and down the country to get access to the data. And that's once you've got all the governance sorted out. So it's the logistics of obtaining those data are enormously difficult. And likewise, you know, if you talk about brain banking, autopsies, what proportion of the cohort are going to have that performed, and the logistics of getting the consent and getting that sorted as soon as the participant has died, contacting the family, how it's actually thinking about doing it on the ground on a day-to-day basis. It becomes enormously difficult, but it's something that we're obviously very happy to discuss, but it really needs to be feasible to be done at a large scale. There's no point doing it in 100 people.
There's a couple of questions over there, and then there's a question over there. So number one.
Thank you. Sarah Gold from Projects by IF. I'm really curious to understand what the panel would like to see in the next year in order for the stewardship of these large data sets to earn and crucially maintain public trust, given that it's clear that more data is needed for better research, and that data will be increasingly novel, as has just been discussed. The opt-outs are becoming more frequent as a result of poor communications or trust breaches. Upcoming changes to the UK Data Protection Bill in the UK is heading towards genetic data sets, and that public expectations are changing in regards to how data about them should be used. And if possible, I'm really interested to hear the panel's ideas that go beyond public involvement, which I believe is the floor, not the ceiling, when it comes to trust. Thank you.
So maybe I could kick off by saying that I think communication with the participants and involvement and engagement of participants is clearly key. So far, the UK Biobank participants have demonstrated that they are very supportive of the project. Fewer than 1,000 of the participants have withdrawn. But we're not complacent about that, and we're certainly starting increasing areas of work around involvement and engagement with our participant panel, but also with various focus groups that have been run and more that will be run. I don't know whether Naomi wants to expand on that.
Well, only to say that, I mean, one of the big differences here with UK Biobank as opposed to other NHS data for health-related research purposes is that we have explicit consent from all 500,000 participants to collect their data through the NHS, to perform data enhancements for research purposes, and we provide the data in a very secure way. But you're absolutely right that, you know, we cannot be complacent, and we take data security extremely seriously. And getting participants involved, particularly around any new enhancements around dementia, is going to involve the participants, their carers. We absolutely need to make sure that they are on board, and that we're doing this for them, particularly if we end up feeding it back to their clinician as well, as much as the research community.
So number two.
I'm Rufus Greenbaum. I'm a private individual. There's a UK charity called Food for the Brain that's tested about 250,000 people with an online test for cognitive impairment, and consistently 11% fail. How does that compare with any other tests that you're currently aware of?
Any member of the panel?
I think it really depends on what the cognitive test was actually assessing. It's very hard to answer that without knowing what the test was.
And I would also add that people obviously doing the test, there's a huge amount of variables. I mean, we've talked about the periods before people develop significant cognitive impairment and activity levels tailing off, maybe as a kind of prodrome to their dementia. But that's a very complex period when people may be starting to get a level of depression, for instance, anxiety about the changes in their cognition. All of these very subtle things can feed into cognitive performance, which again comes back to Siddharthan's comment about why this is just so difficult to get right. So I think it's very difficult to say. I would just say again, from a clinician's point of view, it's maybe not surprising, that level of impairment, but it's probably just difficult to comment in detail without knowing more about the people doing the test and the test.
Yeah. And I'll just add, it's partly the population, but the sensitivity of the test is really, and specificity is really important to ask. So for example, reaction time, which Naomi showed us earlier, was highly sensitive to age, is an incredibly sensitive test to many, many things. Dementia is only one of them. And so one often has to use, well, one always has to use multiple tests in conjunction with each other in order to parse out the specific cause.
So number 4 at the back.
Hi. Yes, I'm one of the original biobank subjects, and I'm now aged 80. 10 years ago, I thought that my memory was starting to go. And my doctor said, can you repeat the 12 months of the year backwards? And because I could, she said, you're fine. Now, actually, things have got worse since then. And I'm now, in fact, Biobank have invited me to join this new research project to see how things are going. But the point I'm making is, nothing had happened over those 10 years. And I don't know if Biobank noticed that I was a potential subject, but, you know, I felt left out, put it that way.
Well, thank you for your comment. And I feel that there is a lot more that UK Biobank could do in terms of communication. One of our issues is the scale of the project is really quite difficult to communicate as well as we would like to. And it's an area where we're really putting a lot more effort in to see whether we can communicate with participants and involve them and engage with them to a much greater extent. But thank you for your guidance on that.
Rory, could I just add, that was 10 years ago. I think the science has advanced spectacularly in the last 10 years, both our understanding of the pathobiology, the refinement of blood-based biomarkers and digital tools. So the field is at a tipping point, and then there's a further impetus because of the successes of these 2 human anti-immune therapies. If you bring this all together, we're at an inflection point, that now is the right time.
And just to add to that, I know Naomi highlighted the number of people who get a diagnosis of unspecified dementia. I think that was data going up to 2021. I would hasten to say that even that has shifted over the last couple of years. Again, having worked in clinical services for a long time, a lot of people used to get that diagnosis, including people sitting in primary care who maybe had the type of level of test you've described. That is starting to shift, but as Siddharthan has said, what's really going to drive the accuracy in more detail are these scalable biomarkers, because we haven't been able to do CSF, PET, etc., obviously on everybody at scale. And there's so much variability in picking people up just with a cognitive test and a structural scan.
Could I just kind of pursue this a little bit? And I was just wondering, maybe I could throw this one at Nadeem just for fun. But, I mean, the comment was made, you sequence 500,000 people, but you still don't know whether they've got what kind of dementia they've got. Can you comment, or others of you comment about how having the genomic data, having, say, proteomic data and other biological assay data might be helpful in combining it with diagnosis, in helping to subdivide diseases?
2 things I'll comment on there. The first is from a therapeutics perspective. Our aspiration across the industry is to develop precision therapies where you understand the kind of molecular biochemistry and pathophysiology causally driving a disease, then you match your medicine to that mechanism. We currently can't do that because we don't really understand all these causal drivers of disease. It was notable that over 70% of life sciences companies use UK Biobank data. And that's precisely why, because it's an unusually integrated resource where you can layer genetics, other omics, and clinical data to kind of guide the understanding of disease processes. So I think that kind of triangle of genetics, biomarkers, clinical outcomes is essential to therapeutics discovery and development. And currently, dementia lags compared to other therapeutic areas when it comes to that. One other thing I just wanted to add related to a few other comments that were made. The UK Biobank has been a pioneer in two key ways for me. The first, of course, is the scalable science. The second is the inclusive multi-sector collaborative models that you've managed to implement, and where you have brought together multiple sectors that frequently wouldn't come together. And one opportunity I see looking to the future is with the digital and tech sector. One thing I recognized is medical sciences, pharmaceutical sciences, life sciences, we're not the experts when it comes to consumer engagement, communication, aspects like that. Other sectors are. So I think one tremendous opportunity for UK Biobank in the future could be bringing together other sectors that we don't currently engage with.
Yeah, thank you very much. So 3 more questions. Number 3 first, then 2, and then 1.
Thank you, Rory. Claire Beamfield, NHS England. You talked collectively about the need for more biomarkers and also the potential of primary care data. I was curious whether there'd been exploration around unstructured data set within primary care. So I'm thinking PDFs, written records, and the role of NLP in that as an additional space that going beyond the structured data within primary care, and what you thought the opportunity might be within that domain.
Yeah, that would be wonderful, but we can't get our hands on the coded data first, let alone the unstructured data. So yeah, let's do it in piecemeal steps. If you can get the coded data, that would be fantastic. But obviously a lot of this richness is written down in free text, and we would want that in due course at some point. But we can't even access the coded data at the moment.
But it may be that if we can get the coded data across the whole cohort, then as with the kind of imaging focus on dementia, one would go to particular, the GPs for particular participants and get that kind of specific information for those individuals. That might be a kind of phased way of considering it. So number 2.
Hello. Some time ago I've always had a very poor memory, so I wanted to have a baseline before I started getting decline, because otherwise I would think they probably would have thought my general memory level was I should be institutionalized. So I went, the GPs have a form, a questionnaire about where they use to judge the state people are in. Now, there is absolutely no possibility that it's going to catch early things. It would catch you when you're at the level of being institutionalized. Should you be institutionalized? To give you an example, one of the questions is, do you know where you are? Now, that is not going to catch early, the fact of not knowing where you are is going to be quite a long way along your decline. That form, but I understand all GPs use this form. It must be rewritten with the idea that now they have to catch people earlier. I'm sure it was written when there was no hope of early interventions, but it must be thrown out, the one that's in use at the moment, and a decent questionnaire, replacing it.
I think you've captured perfectly the transformation that it is undergoing right now. We can't continue to ask people to spell world backwards or tell us who the prime minister is, etc., when we have a much different toolbox at our disposal. So someone asked earlier, what does success look like in a year or in the future? We should anticipate a world where a digital, a blood, and a simple genetic test, before people even realize that they may have the disease, can identify those that have or are at risk of getting the disease. That's a frame shift moment in the way we think about dementia, and that's then a very different way of finding and treating people. And if you combine that with what Siddharthan said, a conveyor belt of novel therapies on the horizon, the way GPs or clinicians diagnose or manage or treat dementia will be fundamentally different over the next generation than it's been in the past.
I think just to say, as you said, the tests absolutely have previously been about detecting dementia, and there's usually probably some other questions you probably answered around that that hopefully are a little bit more sensitive and don't seem quite such a broad scope. But I absolutely agree it needs to change. I think the other thing, though, is we need to be realistic that although we are at an incredibly exciting time with treatment development, there will still continue to be people developing dementia for a good time into the future. I'm very positive that ultimately we will be looking at prevention, but we still need to be able to detect people who have significant levels of impairment who actually don't always come forward and still, think that there's not an awful lot that can be done. And thank goodness we're in a really different place than we were in previous decades when you were just looking at institutionalization. But I absolutely agree stuff needs to change.
Siddharthan, did you want to make a comment then?
It was only a facetious comment because Nadeem said we've got to move away from naming 2 prime ministers. Given the proliferation of prime ministers we've had, I think that would actually be quite a good test.
Final question before we break for lunch, number 1.
Hi, Cornelia van Duin from Oxford, NDPH. My question is in general. I mean, I share your enthusiasm about the blood-based biomarkers and the new digital testing. But how are we going to guarantee in the different settings from GP to UK Biobank that there is a validity test to go on? And what are the standards? Because we want to keep the standards high beyond a simple word testing or a voice test that we had. And I think I would like to hear the opinion on that, for instance, from Vanessa. How are we implementing this?
I mean, I think you're right. I think my comment would be absolutely, as you said, the peoples that present to primary care and come to UK Biobank are a very specific sample as well. And we do need to think that if we're going to understand how we identify people earlier in the stages of these diseases, it has to be in widespread real-world populations, most of which we don't have a good amount of data on at this point. So it is about getting that balance between very scalable, accessible testing that we can implement and also think about how we engage those populations, to be honest, because we know that's another huge issue in even gathering this data in the first place, but then how we validate it as well. I don't know whether others want to comment about plans for UK Biobank around that.
I mean, the only thing I'll say is, Cornelia, I think you've put your finger on one of the things that I think is so important for UK Biobank to grasp in order to play its role in the community, and that is to provide an environment in which these new, highly scalable measures can be validated against strong measures that can be provided in multidimensional ways within the UK Biobank population. So, for example, in the assessment visits, one can use a broad range of digital, fluid, cognitive, and physical biomarkers on subjects who are participating, and then follow them up remotely with the very light touch measures that you were referring to. That provides the cross-validation that allows it to move out into this low-risk population in order to make a difference in general practice.
So I think we've run out of time. I do want to thank speakers and panel members for this discussion. I also want to kind of reiterate the point. We're not telling you what we're going to do. We're asking you what we should do in this setting. And so your response, your reaction to what's been discussed and ideas for how we should enhance UK Biobank for dementia research would be very much appreciated. So we're going to break for lunch now. Lunch is outside and on other floors. If you're interested in learning more about how to use the research analysis platform, there's some people who can demonstrate it to you and talk to you about it. And also there are some posters. And if we could be back in ready to go at 1:30, that would be great. Thank you very much.